So good afternoon, everyone. Thank you for coming. Appreciate you spending your afternoon with us. There is food and refreshments in the back, including water for those that are looking to have a snack. So good afternoon. I'm Mike Kirchhoff, an emergency physician and the Chief Innovation Officer here at Cooper University Healthcare. Welcome to our Cardiovascular and Thoracic Innovation Symposium. So thank you. I'm also our patient safety officer, so you'll kind of get the idea here at Cooper that we're all about solving problems. We made a joke at our last meeting. We don't like technologies in search of a solution or in search of a problem. We like problems and solving problems. And you'll learn here at Cooper that a lot of what we do is in fact solving problems at the clinical level, at the bedside, at the community level, in the practice level, really looking at how can we bring technology and innovation to bear to create better patient care, better experience, better health to our communities. And that's what we're all about. And what we're going to highlight today is one of our core competencies here at Cooper. Cooper's got a lot of core competencies, and we very much want to run at those. Cooper being a tertiary, quaternary referral center for all of South Jersey, level 1 trauma center, academic medical center, affiliated medical school, affiliate with Rowan in terms of having access to all that engineering prowess, et cetera, really uniquely positions us to not only identify problems, but then bring the techniques and technologies to bear to solve those problems. So if you're new to the area, if you don't know Cooper very well, some fun facts about us. 1.6 million outpatient visits, over 100 clinical sites, ED very busy, 80,000 visits, OR is very busy. And we also have an employed physician model. So for those of you who are looking to find clinical validation, et cetera, we're very well positioned from a research perspective to bring in grants, to do research, to manage those projects, but also physicians that are employed and we have the ability to sort of make sure that they have the bandwidth to bring to bear their ideas and create value for our communities and also for Cooper. So really, if you want to talk about Cooper and how we play in the ecosystem that is innovation in Southern New Jersey, Neil and I are available afterwards. So Cooper University Healthcare and our Innovation Center, we're pretty new. I'll give you a little history. So many years ago, our chairman really was interested in creating value for the organization through our intellectual property, but we really didn't have infrastructure. What we were hearing from our inventors was, I have this great idea, but now I want to create value, not only for themselves, but for their patients and the communities they care for. And we didn't have really good partnerships or mechanisms for getting their ideas to market, right? Because in the end, if you have a great idea, if you have a problem that you're trying to solve, and that solution is bringing health and wellness to the community and the patients you serve, we absolutely want to get that out to the community. And Dr. Mazzarelli and our chairman really decided that we're going to double down on this and really invest heavily in it. And that first step was to start the Cooper Innovation Center and hire Dr. Neil Lemon. Neil will introduce himself later on, but Neil brings a wealth of experience to Cooper in terms of tech transfer, et cetera. He's an MBA and a PhD, very smart guy. And what he's really doing is helping us forge the partnerships. And you'll see some of those partnerships here, J&J and ADA and New Jersey Health Foundation, really all the players in South Jersey that are really helping us bring to fruition these great ideas that our inventors are identifying. And for those of you who are clinicians, this was intentional on our part, that when Dr. Mazzarelli thought about starting the Innovation Center in partnership with the Cooper Foundation, one of the central tenets to that was our people are awesome and they should continue to be awesome and be rewarded for being awesome. So we have one of the most generous intellectual property policies in all the Delaware Valley, and we're intentional about that. That's why we want to be innovative. Our inventors bring ideas to us, and we help them commercialize that. And for those investors in the room and those that are trying to help us lever new companies in the ecosystem, what that really brings to the table is inventors that are very invested in their products because they see the value it's not only going to bring to their patients and their communities, but also to themselves. And an invested in and excited inventor makes for a great founder. And we're very intentional about that. So just very excited about the work that we're doing here. So what I'll do is hand it over to Dr. Neil Lemon, who can introduce himself and introduce our esteemed panel. We're going to have a panel discussion, and then we're going to hear about some of the thoracic and cardiovascular inventions that we have in innovation to really drive that forward, and to have a discussion about some of the great things our inventors in the Innovation Center and Dr. Lemon are doing. So without further ado, Dr. Lemon.
Thank you, Dr. Kirchhoff. So I think we're going to try to, we have brought a panel together today to talk about some of the partnerships required to bring early stage innovations that our providers think of to address problems that they see in their practice, to all the way through the clinic and into kind of service here. So through that, I'm going to invite our panelists from the EDA, Mike Riley, VP of Investments, Sarah Beth, please come up and introduce. So we have a few chairs set up for you. One of the focus of this event and the technology presentations afterwards is, being that we're still in February and it's Heart Health Month, that we had a focus on cardiovascular and thoracic innovations. So over the last year, we've been very successful in bringing a number of innovations, specifically on the vascular health side, but also within the other specialties in Cooper, to raise, to get some initial funding for prototyping and proof of concept studies, with the goal of spitting those out and licensing them into startup companies, which will, offer some economic development to the Camden region, but also ultimately be in the hands, in all likelihood, of a final exit with a company like Johnson & Johnson, who's a world-class company that leads in a number of innovations in this space. So that's why I'm very pleased to have this panel here, kind of representing the different partners that are required to move some of our providers' ideas, innovative ideas, into solutions for patients. So maybe I'll have each of you say a little bit about yourselves and your institutions and the role you play, and then I'll have a couple questions for each of you, and we can start into kind of the Cooper technology. So maybe we'll start at the far end with this mics. You've got a microphone?
Yeah. I don't, we need
Dr. Hartsey?
Yes, maybe we need a sound check. Thumbs up. We can try to solve a lot of problems. I couldn't even untangle this this wire here, but thank you for having us. Me personally, I'm Taharji. I'm a pathologist. I'm a physician. I lead the medical affairs, cardiovascular, and metabolic team at Johnson & Johnson. I also lead our innovation team, and I've been in dark side of industry for about 10 years. Started at AstraZeneca, did work for Amgen as well in California, and now local here with Staple Healthcare Company of New Jersey, Johnson & Johnson. The two are synonymous, but I agree with you. We are the largest healthcare company in the world. We take our responsibility to the patients and to those who deliver care very, very seriously. And we constantly look for new ways and innovative ways of incorporating into our business and into therapies to the patient care. So pleased to be here. Thank you for having me.
Sarah Beth?
Hi, I'm Sarah Beth Vaughn. I'm the Senior Life Science Officer with New Jersey Economic Development Authority. If you aren't familiar with us, we are a state agency. What we do is we try to connect, whether you're a startup or you've been around for a while, but to either non-diluted or diluted funding. So we have over 50 programs across the New Jersey Economic Development Authority. We also work close with Judith Schiff. She runs the Commission on Science and Innovation. They have some great programs that we work alongside with them that are non-dilutive funding and grants. And on the other ends, we have, venture funding, we have, angel investment. And one thing that I grew up in South Jersey. I went to Rowan for my undergrad. I work out of the Camden office, so I have a vested interest, obviously, being the proximity, this is home for me. I'm very excited. I was explaining to people earlier, my mom grew up and Camden was a thriving area 50 years ago. And then over time, it's really changed. Then the Christie administration came in, tax abatements, you had Campbell Soup, you had Subaru, you had Lockheed Martin, all of these companies coming in. But then Cooper has been here for so long, but I feel like now is the perfect opportunity to start talking about how do you take these newer companies? How do we work with companies that have funding and institutions and universities, and how do we collaborate to then build upon that? North Jersey, we have strategic hubs that are the Helix and Nest building that are going to be coming online in the next 2 years. But in South Jersey, we don't have that. How do we compete with Philadelphia and then possibly make Camden an innovation hub like they have done in Philadelphia and in North Jersey?
Very good. And Mike Wiley, VP of Investments at FHA.
It's a pleasure to meet everyone this afternoon. Mike Wiley, Vice President. We're a nonprofit foundation that has a seed venture fund attached to it. And we fund projects and companies that are in a nexus with a select group of affiliate partners. Those partners are the major research universities in New Jersey, but also central systems. So obviously Cooper is one of our partners, Meridian Health, Hector Foundation, Rutgers University, Stevens Institute, Princeton University. So any of the projects where companies have to have a connection with one of those institutions, we've been fortunate enough to support a number of projects here. Cooper, and I think we're going to hear from some of the clinicians that back those technologies. Steal thunder, but I'll just maybe back up Dr. Kirchhoff. So it's just been a pleasure to collaborate with Cooper. The innovation is clear. The energy is fantastic. The clinicians, these are well thought out problems that you can tell that they've confronted in their clinical practice, and the solutions are equally as well thought out. So we're excited to back them from a foundation perspective. We're here to improve health in New Jersey and around the world, and so we're putting our money where our mouth is by supporting these projects. Cooper, and looking forward to hearing the presentations later on, sharing some comments.
So absolutely, it really kind of takes a village to move some of these, address some of the problems our providers are identifying and the solutions they're coming up with. We have partners in the audience from engineering companies, from entrepreneurship studios, from different spaces, in addition to the group on stage here. I just want to get a quick question regarding, the mission of your companies, institutions, agencies are supportive of innovation and entrepreneurship, and basically making new devices, solutions, software to address health issues. Right? As far as challenges, kind of putting together that village, what do you find, say, from your perspective at Johnson & Johnson, where you're in charge of innovation and dealing with folks, in the health system or within the universities, they're trying to come up with solutions for these problems and look for you to get to solutions?
Yeah, where do we start? It's easy to identify challenges and problems, right? They're all around us. I think it's very important to find solutions. But for us as industry, the challenges that start with regulatory tape. That's a very easy one. We are a for-profit company. We operate in the healthcare business. We deal in people's health. I always say this is not the place in a toaster. It's a joke, but to me it resonates. And with that comes the ultimate responsibility. So I think the challenges are in many different ways. Excluding the regulatory, I think the innovation space is very robust. And that doesn't just mean technology. a company like ours and an industry like ours dies without innovation. It starts in the bench. It starts with that aha moment, whether it is a device, whether it is a molecule, whether it is a medicine, preparation of any type that can be life-saving for many. So it starts with having the intellectual capability in-house or externally, working with beds of innovation like universities and working with companies that are really robust and quick into identifying the areas of unmet need and really putting their effort and their intellectual as well as finances into that space. Another challenge is to validate those methods. We as many of others, get a ton of offers and a ton of prospects. And you can't do anything with them without proving that they work, they deliver what they say they can, and also that they're affordable. You can have the greatest product out there or the greatest solution if no one can pay for it. It dies at the store. So there's so many levels of complexity, but I would identify regulatory as very challenging. I would identify supporting the seeds of innovation in all different aspects and being able to identify the ones that can thrive and can deliver quick, inexpensively, and also can have the most impact. That they have to, we're in the cardiovascular space, let's face it, there's no question that we deal with the largest problem out there. Pandemics and whatever else comes our way, cardiovascular disease seems to remain the number one. So we have our work cut out for us. We have committed to making an impact into the most challenging health crisis, and I think that that brings some added complexity. The last thing that I'm going to say is the constantly changing political and the affordability piece, the compensation scenarios out there. It's a world that is in constant flux. It's impacted by so many elements. It is always a challenge to have great products marketed the right way and also anticipate a little bit the market for the great product. So it takes a little bit of guessing work, I guess. I'm glad I don't have to, I'm not one of those people. I don't know how they do it, but also you have to really keep up with the trends and the pressures and with all the changing landscape out there in order to be able to feed the innovation. Everything that's gained has to go back in for the next project. So I hope that answered.
Helps quite a bit. I think I'll always try to combine some challenges and opportunities as we go through. I have a follow-up question for you as far as opportunities, as far as methods of partnership. So I know through my career licensing, at what stage is the technology ready to partner with a company like Johnson & Johnson? Is it better to go through and get concept startup a company or a connection on venture side? How have you found it in the cardiovascular space as far as partnerships with technologies coming out of the universities that Mike supports through his investments?
So the easy answer is at all levels. There shouldn't be any area where an inventor or someone with a great idea should hesitate to reach out. We go from the beginnings of the ideas to marketability and scaling all the way to selling the product. So we cover the range. I think we're open as a company, and I do speak for them. We're open to all levels. Something that I would emphasize is for anyone that's considering approaching industry or Johnson & Johnson with an offering or with a project or with an idea, you have to realize that we have the resources, have the capabilities to make that idea thrive as much as we have the ability to say this doesn't fit into our vision or into our current strategy and step away. When it comes to validating from the early phases of technology, we have J&J Labs, just one example. There's so many aspects of Johnson & Johnson that are as tech savvy and as advanced as all the engineers into this spaces are Google, Microsoft, you name it, Amazon. They came from the hotbeds of technology. We have the ability, and we've proven over 100 years of existence to run clinical trials and to truly validate something quick. We have the expertise, we have the patients, we have the connections and the customers, the institutions out there. We do fellowships every year. I know for a fact we get students, pharmacy students from Roland. I know I'm going from one aspect to another, but that's that intellectual property that is just as valuable. So we bring fellows for 2 years that work with us. They learn from us. We learn from them. I think that human component, it's a great opportunity and should not be missed. It builds relationships. It really establishes bridges that institutions like Cooper or others understand better what the company is doing, what the strategic objectives are, where the 10-year or 20-year or 30-year plan is. And then lastly, I would say we have expertise in commercializing. Absolutely, all the machinery, all the experiences there. We do it every day. So I describe those, and there's so many more steps to it, because at any instant, at any segment of that pathway, there can be an opportunity for collaboration. There can be an opening for an introduction. That can be the option for our company and an external partner to work together and make it happen.
Very good, very good. And we'll definitely be reaching out regarding some of our innovation fellows and educational programming to connect Johnson & Johnson with our medical service and bioinsurance. It's a really great point. Sarah Beth, from the life science perspective at the NJEDA, you work to grow the life science industry in the state, particularly in Camden, the office. Working with startup companies, some of the opportunities you see and the challenges you've.
So I guess I'll start out with the challenges. As a state agency, we are the ones offering assistance, so we don't have the challenges. What the challenges are the people and the companies that we speak with. So really, we are supposed to convey what is going on in the market. How are these companies, going from, a idea that was, as Bill brought up earlier, on a scrap piece of paper, to actually getting proof of concept, getting in clinical trials, and then being able to move forward. So really, I feel like in the life sciences, in a week, I could talk to 8 startups, and really just kind of seeing where are they and what are the challenges they're seeing. And then as a state agency, how do we help them? Is it connecting them to universities? Is it connecting them to industry? Is it making sure that really listening and figuring out what their challenges are, and then how do we move forward? Do we need to, develop a new program? Do we need to kind of streamline with, people like from Cooper? So the challenges, I would say, are more or less listening to the people that we are helping, and then being able to implement that and actually have it come into fruition. Not just let it be, oh, okay, these are the problems. No, what are these the problems, and then what are the steps to actually have a profitable outcome?
I can say from my experience with the EDA in New Jersey, they're just expert connectors. So as far as connecting with industry, connecting with funders, you have specific programs as well, which I think are unique to New Jersey. I didn't notice them as much in Pennsylvania, and really well funded, looking to bridge the support with the state, but with also professional investment and with activating startup. So you tell the group a little bit about some of the supports that you provide for life science innovation.
Sure. So Judith, she works with on science and innovation. They have a R&D voucher program where it's $40,000 that for equipment or facilities. And as long as you work with a state institution, so or university. So think about one, you are able to collaborate with a university, but then you're also able to take that funding and bring it towards, working on maybe a proof of concept or equipment that you might need. And then on the EDA side, we have an evergreen fund where we are onboarded 13 venture firms. And in that, we will match the funding up to $6.25 million. So what that is we've onboarded 13 venture firms. From there, people can send us their pitch deck. We then put it onto a portal, and then it is a nice way to have the connectivity with the venture firm and then also with the company. Also, if the company is talking to a venture firm, that could be anywhere in the world, they could be onboarded. So if a company says, hey, I found, a venture firm and they're willing to invest $2 million. Well, if they are onboarded into the Evergreen Fund, we will match that $2 million. So really here in the state, we are trying to take the funding that is, possibly out there, and companies, everyone's going to be investing, but how do we at the Economic Development Authority match that? One other program that we have is called Founders and Funders. I would call it like a, matchmaking, round robin dating kind of aspect, where in a day, a startup can meet between 10 and 12 venture firms, and they go from table to table. You are matched with the venture firm based off of what your expertise is. They're not going to match you with a venture firm that is looking to invest in therapeutics if they are looking for, med tech. So in that sense, we have run it twice a year, but it's a good way for companies to meet a face-to-face, have a warm introduction with that that venture firm to then move forward in a direction of investing. And then those are just two. We have lots of programs. And I can give you my card. We can talk further, but
Absolutely. I know here in Camden, we have some areas of focus where Camden's really like a national or even a world leader as far as healthcare and some of the technologies. So we've had discussions as well about different opportunities for innovation centers or something like Camden, which the NJEDA also plays a role at the state level supporting. So ongoing discussions and hopefully some good news coming up in the future for that. And Mike Wiley and the Foundation for Health Advancement, absolutely instrumental to the success of a number of the technologies that are going to be presented in a few moments here. You have a bird's eye view on health tech development across the state, across institutions, be they Princeton, Cooper, Rutgers, Hackensack Meridian. What are some of the underlying challenges that you see as you engage with, inventors looking to make a difference for their patients?
I'll put it maybe in two categories. So I think on the university side, you're a faculty member there, tenured faculty member. You're not likely to leave that tenured faculty position. And you may be a basic research scientist. You may be world-class research scientist, but maybe you've never commercialized any technology. So a term we like to, and maybe I'll just say on the health system side, it's a similar situation for a clinician who is an expert at what they do. They're unlikely to leave their employment as a physician to go full-blown into commercializing the technology. The term we like to use is we try to meet this inventor or entrepreneur where they're at. And some of them are serial entrepreneurs. Some of them are very skilled or want to be really involved in the company. And so they're actually easier to partner with. For the basic research scientists, if they have a great idea, then we bring basically venture studio type services to the table. We handle the back office. We have a team of venture associates and other team members within our organization that can do everything on payroll, HR, you name it. We can help basically bolster these teams so that they can take that leap of faith in terms of bringing this forward. I think that's been some of the challenging side is you have a great idea, maybe the university files a patent, but there's so many steps from there, especially in healthcare or any other regulated industry. There's so many steps that you have to undertake, so it almost seems overwhelming if you've never experienced it before. So we try to just kind of flatten that learning curve and provide them the resources to make it a little bit easier. So I'll say on that, there's some challenges, but we're trying to bring these opportunities to the table. And we don't do it ourselves. We partner with external consultants, other organizations, people like J&J, other folks within industry to help commercialize these technologies. In terms of our venture fund, we like to fund that pre-seed, maybe seed stage of development, but really that's to de-risk the technology. So other venture capitalists, industry partners can come in, and that technology kind of fits their paradigm. Because a lot of times when it's first coming out of the institution, whether it's Cooper or a university, it's just still too raw. And a lot of industry players or even Series A venture capital funds won't make an investment at that point. And so we don't want it to languish or get stuck in the laboratory. We're going to make that pre-seed or seed investment, get through a couple of milestones such that an outside investor or industry partner can come on board and start to move.
And it's an instrumental position, like that first money into a venture. You know, my time before I was at Cooper, I was 5 years at UCI, and there was like a very limited number of venture groups that would form the relationships that early to be the first money in, and then eventually it comes out. You know, Series A is syndicated. It works out. I know visitors from MBC Studio as well, who work in that space on the med tech side, device side, also looking at initial non-dilutive funding in, first money in, what partners can be put together. What I love about the program that the FHA FBCG has done is they've also operated with fellows. We have Troper here, one of their fellows, 4 different fellows from that organization. And they put the sweat equity into the company, and that sort of work that would otherwise need to be done by a management team, which you can't necessarily attract into the venture at this stage. And, you know, they're experts. Mike Wiley's one of the most plugged in people in the health tech space I've met, definitely in New Jersey. And the introductions and helping structure how the technology should look and how it can grow. And also validating some of the issues typical of these startups. Very, very important. Just one last quick question and get to the presentations. From your perspective in New Jersey, Mike, what do you think is a couple of the most exciting developments? I know there's several where there's been investment in, and a lot of companies that have kind of been looking at have New Jersey stand out as the top of the life science pile.
Yeah, I mean, I just didn't think the life science enough to comment on this because there's so many infrastructure related projects in the life sciences actually around the state. It's really exciting. So we're here in South Jersey, and just this morning, more is coming our way. Super excited about what's going on with Cooper, but in the broader South Jersey area. But there's equally up in North Jersey and City and some of these other life science centers. We're seeing a lot of infrastructure coming online. And then I think we all look at what's happening on the health side related to AI, and whether it's in drug discovery and other areas. And I think New Jersey is actually very well positioned to marry kind of our core life science and clinical insights with some of the IT history that we've had and some of the companies that are resident here. So I think we're going to be able to leverage some of the new technology that's coming down the pike, and we're going to have the infrastructure to take advantage of that. I have a huge legacy with companies like J&J, and I know they're leaning in on some of the technology. So I think we're extremely well positioned in life sciences.
I'd like to just make one comment. My commission had previously funded STEM Plant, the company Spencer Brown. They're from, and I just wasn't able to quickly find it enough on my phone if it was an SBIR.
It was an SBIR, phase one.
Matching grant that I had been able to, to give them. So we have examples where we have funded work that came out of collectively this team.
Yeah, and STEM Plant is a Cooper startup focused on stem cell delivery to the vasculature. Made some prototypes. We worked with the company, with the piece there. Really happy with that, Spencer Brown, and we're continuing, actually hoping to do some prototype testing in the coming couple of weeks.
So if he's doing a prototype using some of your facilities or university facility, 40K on that voucher that Sarah Beth talked about. We can talk later.
So I'd like to thank the panel. Thank you for coming here. We're going to have 15-minute presentations from our inventors, clinician inventors, and we'll leave about 5 minutes for questions afterwards. So please take your seats and feel free at the end of the presentations to hands up and help field questions. And so the first presentation is from Dr. Barty. I should say nearly all of these technologies, I think one so far is still in the preliminary stages, have been supported through the Thanks.
Good afternoon. Thanks again for this venue. And I'd like to take a moment and give a special shout out to Cooper. And I think, as a surgeon, you're really busy in the operating room, really busy taking care of patients. When you go home, you really have to try and catch up to life. And there's very little room for us to start thinking about innovation. It usually develops in the OR or some area, and you're really busy, and you're like, what if I just had something like this? And then it disappears. I think what Cooper has done is given us a process and a pathway that really breaks down all these little barriers and all these little mental barriers where you think that you can't get an idea across that threshold to where it's involved in some deliberate development. And they've really done that for all of us. And it's special to me because it's been such a fun process. It's also cascades down among our learners. And half of my division is here today listening. I'm talking about our residents and our nurse practitioners and EAs. And thanks for coming, by the way. And you can tell that this fever of innovation really can catch hold. And Cooper really has done a great job in hiring people like Neil, who's been fantastic at this process. So thank you so much. Our friends at Jersey Health Foundation have been fantastic in this whole process. So although we're kind of still crawling with this current concept that I'm going to present to you today, there is a clear plan articulated for flight. So to that, I'll talk about what we have developed over the last year using these resources and opportunities. I'm an aortic surgeon, so I deal a lot with aortic aneurysms. And the type of aneurysm that we're looking at here far on your left is a thoraco-abdominal aneurysm. It really starts up in the chest and ends down in the abdomen. And classically, when we repair these, it involves a really big incision. And there are elderly patients with a lot of comorbidities like chronic obstructive pulmonary disease, coronary disease, peripheral vascular disease, whatnot, which is the typical milieu of these types of patients. And what we're trying to do over time is minimize the amount of insult to these patients that in an effort to fix exactly what they've presented with. So you can see on the right hand of your slide, there are these iterations of what's called endovascular devices that have been implemented to minimize the toxicity of these big open surgical procedures. And there are variations of these technologies that have been implemented and available, scattered throughout the United States. Widespread availability in Europe, I will say. They are first to market with almost everything in the past. Not so sure that that's a future attribute for them. But what we're looking is to minimize either the incision, the exposure of the patient, and the length of surgery required for these patients. A lot of these patients require cardiopulmonary bypass, so we have to literally put them on a heart-lung machine to really get them past some of the visceral or arterial reconstructions that are necessary such as what you see here. All of these little important branches go to vital organs, and these need to be perfused while you're repairing these aneurysms. So this is kind of just a 30,000-foot view to take a look at the context and difficulty of what we're dealing with. And what we've come up with in lack of suitable technology is custom making these devices ourselves. So this is us in the operating room burning holes and if commercially available endograft, fitting them precisely to the patient's anatomy. So all of these are very, very well-defined measurements and holes that we create in these grafts, and we sew these rings around and pacify them with wires that you can see on fluoroscopy. And we implant these, we resheathe these and reimplant them into the patient. And we've had some stellar success, but it really shows you the gap we have with taking solutions to patients that are commercially available. These are not commercially available devices. So we have to sometimes be innovative and try to overcome some of these challenges and make these patients whole again in a minimally invasive fashion. There's about nearly 63,000 diagnoses of abdominal aortic aneurysms last year. In 2019, 80% of those were repaired through these endovascular means. 125,000 abdominal aortic aneurysms and thoracic thoracoabdominal aneurysms were performed globally, and aortic stent graft business is set to reach about $4 billion by 2024, and it's here and now. So the projection is going to continue to rise, and we're going to continue to develop innovative ways of tailoring these aortic stent grafts to patients. Now, what we're really looking to do with this technology that I'm going to present is break up this procedure into two steps. We're going to do a minimally invasive procedure, which is what I'll call a TEVAR, thoracic endovascular aneurysm repair. And that is placing a graft, as you can see here, because my pointer, yeah, you can see it. So putting a thoracic graft, and this literally takes about 15 minutes to do in an office, to deliver this stent graft. And then close the groin with a minimally invasive device that plugs the hole, and then you have the patient go home. Then to come back and have a not so minimally invasive visceral reconstruction where the patient can have an open procedure with a more limited incision, and you can reconstruct the visceral vessels as we see here. Now, the benefits is that it eliminates for this big surgery requiring a large incision and getting these patients usually in the intensive care unit for about a week before they're able to come out. And it allows it to perform in two stages. The spinal cord can have some time to adapt because spinal ischemia is a big problem with this type of procedure. And once a patient develops paraplegia, they have a 50% mortality. So this is a big actor and a big consideration when we think about these cases. And then you have patients that come in with the challenges of still requiring cardiopulmonary bypass even for the second phase. It still has a lot of time-consuming visceral reconstruction, and dealing with the endograft distally can be very challenging. So here you can see there's a little graft on the inside where a TEVAR was placed. You can see this aorta is aneurysmal, so we kind of have to bunch it up and tailor it down to the right size, and then fashion a graft where we sew this graft to the endograft that was placed before. It should look like this, but it doesn't quite look like that on the right side, does it? So this is a challenging part, and meanwhile, you're on the clock. So these important blood vessels that I pointed out earlier are without blood flow at this point. If you're on cardiopulmonary bypass, well, that's about an extra hour to set it up, and the patients are really run thin on their anticoagulants. So their ability to make clot before and after is brought to a real minimum. And that gives us the opportunity to clamp without producing clot. So cardiopulmonary bypass is a big deal for these patients. So even with this second phase, you need cardiopulmonary bypass to alleviate the stress of trying to do everything on the clock. Now, There are a lot of publications brewing with respect to hybrid repairs, and this is what I'm going to call hybrid repair. And here's a group of friends of mine from the Cleveland Clinic who have published on this type of repair and had some excellent results. And as you can see, the popularity of these types of cases have really gained traction, particularly at centers with high volume like the Cleveland Clinic. So this is here to stay. This is here going to be a mode of repair that is very well accepted among large centers who do a lot of open thoracic reconstructions. So the second stage problems, again, we talked about the cumbersome graft. We talked about hemostasis for patients being on the pump. We talked about the visceral ischemic time. And these are all things and problems where we say, hey, wouldn't it be cool if we had this? And our solution to this is coming up. It's to treat these zones 3 to 10 of the thoracic aorta. These thoracic aorta is broken down into zones. And it involves visceral involvement where all these important blood vessels going to your important organs lie. And as a continuation or as a de novo product for thoracoabdominal aneurysm repair with or without TVAR. And TVAR is more likely to be performed prior to this second stage. So this is a second stage item. And the second stage item is an endograft, again, much like the TVAR, but it is fashioned along with a visceral open segment reconstruction Dacron graft. And here you'll see that fully expanded, you have an endograft that is sewn onto this Dacron graft with branches that are supposed to mate with the important visceral vessels. The key kicker here is this perfusion cannula, this little veil that is supposed to be sewn to the aneurysm. Remember I said the aneurysm is really big and you've got to kind of bunch it up. This eliminates the need for that and to control hemostasis. And then again, you have this perfusion cannula that hooks up to this very convenient manifold. And the manifold hooks up to the perfusion cannula. And this is after you clamp the device right here. A clamp is placed after the device is deployed. The balloon is expanded for hemostasis. This is connected to the manifold after a clamp is placed. And then I have the ability of taking this manifold and selectively placing them into the vital orifices of the visceral vessels. Now I could just relax. We don't really ever do that. But hypothetically, we can sort of just alleviate any concern that the patient has ongoing ischemia from this clamp. And the interruption is temporary, but it allows us to selectively take care of each visceral vessel in a very controlled fashion. There's no bleeding. There's excellent perfusion of the downstream vital organ systems, in addition to a potential solution for the distal aorta. And then this is it in its full glory, with all the perfusion cannulas selectively inside the visceral vessels while you're taking one visceral vessel at a time while the others are being perfused. And then this is what it looks like at completion. All the little branches are sewn onto their respective visceral arteries, and then you still have what's left of this perfusion manifold that is taken off, and that little area is just sewn off and tied down. And then you can worry about the distal aorta, whatever reconstruction that needs to be made. Now, I'll tell you, any aortic surgeon who sees this goes, son of a bitch, why didn't I think of that? And I'm being a little bit of tongue in cheek here, but this is so sorely needed, so sorely needed. And I can't believe it hadn't been developed. It won't have a massive market, but it's going to have a. And I think that this is something that will help aortic surgery really take the next step innovation. Our future projects are to take this perfusion manifold that we have, and for emerging cases, you're not going to have this. You might have this off the shelf, but you might not. But when you have a rupture, and the rupture involves that visceral segment, it's great to have this and basically just stick it into the aorta above where you clamped on the sheath and allow that blood flow to temporarily give you a bypass to your visceral vessels while you're doing the visceral reconstruction. There's nothing on the market for that. And we're currently developing both ideas in parallel to offer some solutions to these types of scenarios, which occur a handful of times a year. And again, it's a disposable item. It's not an implant. It should be easier from a regulatory standpoint to get through the maze. But that's it in a nutshell. I'm happy to take some questions. But again, thanks all of you for your time today. And again, thanks to Neil and everyone else who's been so helpful.
So who would you envision making the device? Is this something, that a stent company already, you would license ultimately the technology to them and they would make it that you would?
That's an option. I think most companies today, like what was said earlier, like to see a product that's a little more well-developed, a little more that sweat equity into it, a little more early investment, and at least to the point where you have some big models and prototypes kind of worked out. But that is an option to present after that phase. And then there's also the opportunity for equity firms to get involved and build their own little business. And I like the fact that we talk about building your own little business. And I think that's the plan until it's no longer the plan.
So you'd want to probably think about what other products, so that I'm not just a one product company, but what's kind of the range of other capabilities that your company would be offering.
Exactly. And, that temporary shunt concept can go in many different directions. So we're excited about that as well.
One thing we have, or I've seen over the last 10 or so years, looking at licensing technologies as well, is like once you develop, you get a prototype proof of concept. Quite often, in acquiring licensee wants to see first commercial sales too. So we're in a position to do that through partnership with PHA, FMCG, with their support. That started up as a business, and if it is a one product company or if it's a two product company, kind of so be it. We're going to make sure that we get it into the clinic and get it to the point where we can have an
Just one other, it's a very good point. We're certainly, that's an exposure that we're concerned about. And you'll see, and that's why I mentioned some of our core competencies here at Cooper: endovascular, vascular, neurovascular. There's a lot of vascular. So we have a suite of solutions. You're going to see some other catheters. We have other catheters and grafts that are in the pipeline. So the idea is also to not only run out our core competencies, but leverage those core competencies in a way that we can create a suite of solutions. And that suite of solutions then becomes more attractive for acquisition. So still that same prototype, animal models, get some clinical data, et cetera, but also understand that we have this valuable IP. Where else do we have similar valuable IP that we can then bundle together to make that suite of solutions more attractive to an investor or a company? And we're very intentional about that, because exactly one-off solutions, the pathway to commercialization is not quite as clear.
So Mike, it's making me think of polymerics. The Rutgers company from, I don't know what, 20 years ago.
An example is, like, say, in a Symposium, we have a few really, really interesting vascular health company that has an interest in one of our startups, or directly with the guys with the initial assistance of the FDA. So,
That's another question. Is there a specific thing that when you're doing the work in the search, like you have to customize it? So is there like another device that you might be making to make the customization of the, maybe I didn't totally understand. Is there
No.
something that you're doing in the search, like almost like,
Yeah.
like you have a little makerspace thing that you've got to do in the surgery suite to then stick the thing in?
With this, you probably won't need to do that.
Okay.
This offers a lot of versatility. You don't have to connect all 4 vessels, maybe you only need 2. Maybe you don't need a T-bar ahead of time. You can just use the graft itself to fix it. So it has a lot of versatility with respect to certain types of clinical scenarios that can be presented.
Okay, so I have a physiology question. So if you use this device so far, Have you seen a reduction in acute kidney injury?
Well, that's a great question.
Thank you.
So we'll see whether or not, when we go to prototype, we're going to design these perfusion cannulas to really be what's big enough, right? So I mean, our diameter is going to be directly related to our flow. And what is the optimal flow? And can we put something big enough on that delivery system that'll suffice? These are some of the questions we still have to answer. So we'll be very in tune to that question. Thanks for that.
Okay. Thank you, Dr. Lombardi.
But
Dr. McMacken will be presenting her technology that she has with Dr. Carpenter. Thank you for coming out today. You know, Dr. McMacken is expecting, so very much appreciate making. That mic should be.
This mic should be good enough. All right. Thank you all for having me today. And as Neil mentioned, I am 9 months pregnant, obviously, and fighting a little bit of a cold. So bear with me if we're taking breaths in between slides. I have very poor pulmonary reserve at the moment. But I'm Catherine McMacken. I am not a New Jersey native, but I've been here over a decade. I was actually part of the inaugural class of medical school and have been here ever since. So innovation began right across the street. I'm working with Dr. Carpenter on this project, who's our chairman and chief of the Department of Surgery. And just as Dr. Lombardi talked about, this was an idea that came up in the OR. The two of us were working on something, struggling with something, an hour and a half into it, thinking there has got to be a faster and more efficient way to do this. So we came up with the bidirectional access device. And what we're going to be talking about today is the background in the vascular space, which is very different than the vascular space that Dr. Lombardi was talking about. He's talking about the belly, he's talking about the central system, and I'm talking about everything else. Your arms, your legs, your neck, everything else on the highway. I will talk about the problem we ran into, how our bidirectional access device fixed that, our market, and then where we are in the current stage of development. So today we're talking about the angiogram. This is a minimally invasive way to diagnose and treat diseases of the blood vessels. That's everything you could possibly think of, from your legs to your arms to your neck. And you can see here on the right of the screen, that is what the picture looks like. You have nice, beautiful, tapered blood vessels, and then ratty sections of blood vessels where you have a lot of disease. Your blood vessels throughout your body are basically just a giant highway system. Everything's connected, but you need to get an on route or an off route. And all vascular procedures start the same 5 steps. So you got to create your on-ramp. You got to create your connection between the outside world and your vascular system. So the first step is you put a needle from the outside world into your blood vessel. Through that needle goes a wire. The needle comes off. And then step 4 is a sheath goes in. Now, step 5 is all of the things you put through the sheath. And step 5 is where most of the innovation in the vascular world comes from. Everyone's thinking, what can I put through these sheaths to treat the problem down the line? You can put in balloons, stents, you can freeze things through this. You can put in ultrasound, all of the things you may have heard of in the vascular space. Our problem is actually focused upstream. It's step number 2. And it's something that is so innate in a vascular procedure that people just assume that this is required. And what that is that vascular disease is unidirectional. Once you put that wire in, you're either on I-95 North or you're on I-95 South. And you can't get off and go the other way on the highway without sticking again. But vascular disease is not unidirectional, it's diffuse. If you have disease going down your leg, you probably have disease going up your pelvis. But the person who's doing the innovation has to pick I-95 North or I-95 South when they go to do their procedure. For example, if I stick the right groin going north, I can treat the arteries of the pelvis and the arteries of the left leg. But if I need to go down that right leg, I either need to stick again on the left side, having multiple procedures, bringing them back a second time, or stick them that same procedure, doubling my #1 complication rate, which is an access complication. So 5 to 8% every time you stick a blood vessel, you're going to get a complication. That's either bleeding or clotting. So you're either hemorrhaging or you've cut off the blood flow to the leg. And as vascular surgeons, That's probably our number one call at night is vascular surgeons, cardiologists, interventional nephrologists, interventional radiologists. Everyone is accessing this highway system. When you access the highway system, you have the possibility of complications. The other issue is, let's say I'm accessing that right groin and I want to go down the right leg. I don't want to go up over the pelvis and around. It's just not ergonomic. It's not ergonomic for a couple of reasons. One, the imaging system that's emitting all of these dangerous x-rays is now closer to the person doing the procedure, which is not ideal. You don't want to be irradiating yourselves during this procedure. The second is you're trying to fight against a pannus. So the belly, as a belly, is reaching over the groin, and you are trying to get it going down against that belly. To give you an idea, if it's hard to visualize, this is actually from one of our cases. And you can see 3 hands here. There's 1 hand here, 1 hand there, 1 hand there holding up the pannus. None of these are the hands of the patient. All of these are the operator's hands, the assistant's hands, being irradiated while trying to get this access because it's so physiologically complicated. You can see down here, this is the stent they're trying to intervene on. Here's trial wire number 1, going some off the wrong way. We have no idea where that wire is going. Here's trial wire down number 2. That's going correct direction, but now we got to try to get a sheath going that direction. So it's just not ergonomic. And the problem's not just in the legs. So this is a dialysis access graft. You have to stick it twice. Same reason. You need to be treating I-95 North and I-95 South. You got to treat inflow and outflow. You got to stick it twice, doubling your complication rate, and also doubling your cost because now you need 2 sheaths instead of 1. You need 2 wires instead of 1. You need 2 booms instead of 1 going each direction. So this is our solution, and we'll have some fancier 3D CAD models further in the procedure, but this is what we came up with after the OR, the bidirectional access device. And this is how it works. So here's your blood vessel, head to the right side of the screen, feet to the left, and we're on step 2. The wire's gone in, the needles come out. Our device comes up over that wire. Once it enters the blood vessel, there's this little depth indicator that bleeds, tells us exactly how deep it is. We deploy this foot plate with this lever. Foot plate deployed, pull back against the artery wall. And now we have wire access going one way. And we can enter our second wire that through a hole in the foot plate goes down I-95 South. We push against the artery wall, make our foot plate flat with our device, walk our device out over our wires, and now I have access I-95 North and I-95 South. 1 stick, 1 access site, 2 different directions. The second part of this innovation is also a sheath that can especially be used for antegrade access, meaning access on the same side of the patient. This sheath has a straight part within the body, which is characteristic of almost every sheath you'll ever see. But then the sheath outside the body actually curves away from the patient, allowing you not to be working right underneath that pannus, allowing you to be working further away from the table. People sometimes try to create these with straight sheaths and hemostats all over the patient to try to bend the sheath, but they're just not made to do that. It makes it much more difficult to push because you don't have a fulcrum to push against. This sheath gives you that fulcrum to push against. It brings the surgeon and their assistant further away from the ionizing and dangerous radiation, and allows you to work in shorter distances because you're going down the same leg instead of going up, over, through the pelvis, and down that same side. So what's our peripheral market? These are some older numbers. I'll have some more updated numbers, but everything in medicine is going minimally invasive. Vascular, the nephrotic, the interventional cardiology, all of that is the same with pushing towards minimally invasive. This past year, 850,000 peripheral only minimally invasive procedures. They reimburse somewhere between $10,000 and $25,000 per procedure, depending on what you do. The dialysis market, 330,000, and they're somewhere between 1,800 and 5,000 per intervention. So the total angiography market outside of cardiac caths is somewhere around 10 billion a year. It's projected to grow to 18 billion by 2028. So if you look at where we think we fit in these spaces, the peripheral market, this would be applicable to about 20% of your peripheral market, but a lot higher percent, about 45% of your dialysis market. If we can capture 25% of each of those markets, that gives us about 109,000 cases a year where we think this device we could capture. Based on comps, and I can go into the pricing later if you'd like, for the device and the sheath, we're looking at somewhere between high 80s, low 90s in terms of a market per year for our device specifically. So we had the idea, we've protected our intellectual property, and then with the help of the New Jersey Health Foundation and Synectic, we're in prototype development. So we've been fortunate enough, we've done component testing, worked through some issues with the foot plate, we've done some low fidelity models, and now we have locked down our final model. There we go. And the final model, thankfully, is being delivered to us in March. So we expect that to become mid-March. We've already engaged a regulatory consultant to start, which we're hoping is going to be a 510K pathway. Animal trials should be early April. And then we're not going to make the March SBIR, but we're looking for the fall SBIR. A lot about blood vessels all at once. Happy to take any questions.
Just please, after you, sir.
Could you go back to the slide where you introduced that blocking device, the blocking slab?
This part?
Yeah,
Yes.
where'd that belong? Blocking device.
So this foot plate is actually part of the device here. And this, the foot plate in and of itself is not innovative. The foot plate with the track through it, through which you can pass a wire, is innovative. So the foot plate that comes out, that you pull up and hits the artery wall to tell you where you are, that's already integrated into a variety of vascular devices. So when you put that part of this in someone who does interventions hand, that part they understand because they're used to pulling up a lever, pulling back, and having that tactile feel of where you are against the blood vessels. Some devices use a foot plate, some devices use a balloon that you inflate and then you pull back. But that's a very intuitive feeling for us of I'm in flow, I pull back, I'm up against the wall.
So your plate has an opening in it, second wire?
Correct. It has a channel through the middle that's contiguous. I have a 3D CAD.
There's the alignment with the.
If I go through, no, so the existing wire and the new wire are never in the same channel. So the existing wire is something called a monorail system, where it's in a completely separate channel.
Yeah,
Let me
no, I
see.
know there's something you're checking.
See if I can find my 3D CAD. Here we go. So the initial wire, this is all actually within the device itself. The initial wire comes through this anterior portion of the device. When the foot plate is deployed, it goes through this channel here and then comes out through the foot plate. So the foot plate, one side of the foot plate just has a hollow channel that when you flip it up lines up with a channel in the middle of the device. The two wires are never within the same.
Very good. Yeah, there was a significant trial and effort with the engineering partner, given the stringencies of this. It's very excited for the testing.
Oh, yeah. No, fantastic. As always. The one thing that this slides didn't show is the cost of these complications that can occur, which are really profoundly significant. So access complications can really incur significant costs to the health system. But just with one patient can be upwards near hundreds of thousands of dollars. So that's something to consider as well. And I think a big driver for a hospital to introduce something like this into the system.
And not just our in-house complications, but we have people flown here from all over South Jersey for access complications somewhat regularly. So that is a huge strain on the health system.
Thank you very much, Dr. McNaught. Dr. Mulligan and Dr. Good. We can use both microphones. Yes, we just have one.
Hello, test. Oh,
Hello. Okay. All right. So while we're getting prepared, a little introduction, so I get an extra minute. So Dr. Hirsch has been working on this long before I arrived at Cooper. But when I interviewed at Cooper, they said, you know, Dr. Hirsch is really going to need your help. So I said, who's Dr. Hirsch and what does he need? I'm going to take you through the tale of how we became friends, sometimes enemies. So he had this idea, and I'll give you some groundwork after the completion of the introduction. Evaluation of cardiac function is generally always done today by echo. Long ago, when I was in grad school, we did this invasively. So to get to echo, there have been huge strides made to create the tools to do everything non-invasive. Non-invasive is not always as precise. In fact, it is never as precise as invasive. Invasive is irrelevant today. Okay? So studies that I participated in the 90s and the 2000s when I was at Medtronic, we wouldn't get an IRB to approve today. And they were just simple. Okay, so here's what we have for tools, non-invasive and invasive. So this is what's on the market or coming to the market. So we have lung impedance, which has been around forever, and it tells you about congestion and pulmonary capillary wedge pressure. Echo, of course, has gone from EF to strain rate. Strain rate is the new child that needs to be reined in. Then we have CardioSense. Came out of the Georgia Tech, and they've proven that they can estimate pulmonary capillary wedge pressure and get a sense of stroke volume and contractility. And then the invasive, which we are never going to do, is a Millar catheter in the LV, or combined now with a conductance catheter, so we can do PV loops, or as is done here regularly, we use transesophageal echo during CABG and other procedures. We get EF strain and strain rate. And the old but goodie Swan-Ganz catheter. So the population of people today that could benefit from what he's going to share with you are as following. We have the heart failure population with a very high number, huge costs, and now, for about the last 10 years, we have the presence of heart failure with preserved ejection fraction, which has nothing to do with heart failure with reduced ejection fraction, and it is primarily in women over 50. And it has no FDA-approved therapy. Then what else can we look at with this device that sits on the chest? We can look at how obesity impacts cardiovascular disease, right? So we now know we have an epidemic in the US, and this is primarily due to the sugar taking over when fat was eliminated from the diet. And then the next population that could benefit from this device and the metrics are those patients who are developing breast cancer. So we have a study that we're going to start soon to look at this. And then we have the diabetes population. So this is not just about heart failure, it's about cardiovascular disease, right? Dr. Lombardi talked about all the comorbidities. So a little fundamentals. So on the left, we have a Millar. So this is from a human study in 1998. So we have a catheter in the LV, we're measuring LV pressure, and we're measuring the rate of rising pressure. What I want you to pay attention to is that the black level lines on the left go up as heart rate goes up or dobutamine is infused. And on the right, it's flat. This is a hallmark of heart failure, and it's a lack of the ability of the heart to respond to chronotropic or sympathetic activation. And in this population, you could see that as heart rate went up, both in the controls, there was an increase in the squares versus the circles. This is with dobutamine. And in the 4 heart failure patients, they were flat or negative. Now we're getting to the link to Dr. Hirsch's tool. So LV pressure tracing with the PDT, systole, positive peak, diastole, the negative peak. These parameters do provide a very sensitive measure of the contractile state of the heart, albeit they are dependent on preload. So on the bottom is how these two events are aligned with Bob's tool. Now Bob gets to take over.
Thank you, Larry. And we are friends. So, so I'm Bob Hirsch, and I invented this approach, or if you will, or discovered, I'm not sure which, this approach to measuring heart function in systole and diastole, in systolic and diastolic heart failure. And what this consists in is looking at two streams of data. One is your ordinary lead-to EKG, which I'm sure everyone in this room is familiar with, right? You know, P, Q, R, S, T, right? Okay. And then there's something a little bit novel. It's called a seismocardiogram. That's seismo, as in earthquake, because every time your heart beats, there's a little earthquake going on inside your chest. And in true Newtonian fashion, there's this action-reaction thing. Your whole body jerks back and forth, but you can't feel it because it's very, very tiny. But a very sensitive accelerometer can feel it and magnify it and graph it. And there you see, right over here, this is the seismocardiogram. And I didn't make it as big, but what I did was I took the seismocardiogram and I did calculus on it. Remember calculus? Right? You differentiate things, you get a derivative, rates of change, slopes. This is the derivative of a seismocardiogram. Here's the EKG underneath. And I discovered something very interesting. If you take a signal event in the EKG and you see how much time the concomitant event happens in the seismocardiogram, right? There's a little time delay there because there's this magic thing called transduction. Calcium, you know, moves out of one compartment into another inside a cell, and all of a sudden fibers start moving past each other, and then the whole thing happens in reverse a few milliseconds later. It's miraculous. It keeps happening until you die, right? But by actually measuring the time that transduction phenomena happens, taking its reciprocal, you now have a speed of which characterizes the transduction and the detransduction, which happens every time your heart squeezes and relaxes, right? You have a lightning, here's the lightning, and you get the thunder. The lightning always comes before the thunder, and that's just the way of the world, okay? So by measuring 1 over this interval between, this is the second derivative maximum of the EKG. This is basically a Q wave. You get a ratty looking Q wave here, but look, in the second derivative, you get this massive spike, a signal which rises way out of the noise, right? So from this event to this event, this is the electrical mechanical inotropic interval. Conversely, the T wave, the much more subtle activity or event, but you can see here, at some point, the T wave starts to rise out of the noise, and it rises very quickly. And here's the second derivative of the T wave, and there's a little peak there. Okay, that's the signal. That's the lightning. And then follows the thunder. So by taking 1 over these lightning thunder intervals, these electrical mechanical intervals, I have shown, I have discovered, I have proven, to my satisfaction, maybe not to all of yours, but I'm pretty sure on this, that 1 over this time interval is linearly proportional to the logarithm of the strain rate that you see on a 2D echo speckled Doppler device, which costs a fortune, requires a technician and a cardiologist to interpret. And the other problem with the 2D echo is you can't wear it. You can't walk around town. You can't wear it under your clothes, right? It's not a monitor, people. It's a diagnostic device, right? In my hand, I have a gizmo called a MoveSense. You can see it's about the size of a quarter or half a dollar. Half a dollar. And it's got 2 little electrodes. These little silver thingies on the back, those are the electrodes for a 1-lead EKG, like the one I just showed you, right? And you can wear this with a strap around your chest, and you can get these 2 data streams coming at you, and you can have them Bluetooth to your phone, which has an app called the MoveSense app, and you can buy this thing for $100 from a guy in Finland who manufactures them. And then you can create your own software platform on top of this hardware platform and create a company around it, which is what we're going to do. Right? And we're going to sell it for American money and see if we can change the world. So the
You want some help with this?
you help me with this one. I can do this one. Yeah.
Since I made.
Larry made these graphs, but
Okay.
yeah.
So we have baseline data. So this is strain rate. Sorry, isn't my voice bad? And then we have dobutamine at 10 and 20. Now, we did use doses to 30 and 40, but not every subject had a good signal. So we want to do paired analysis. We're sticking with everybody's got 5 data points. Every patient's got 3 data points. So what we see is as the dobutamine 10 mics, it causes the strain rate to improve, which is what you expect. This is kind of like the PDT that I showed you in the normal subject. And then we go to 20 mics and it continues. So if you go to the diastolic strain rate, you see the same phenomena. So the signal is aligned with what we can measure non-invasively, which is echo. And so I did all the stats. So I tell you that when I first did this, I thought I did it wrong because I never would believe there would be 3 points with a line like that. So I did it again. I started from scratch. I entered all the data again. So the R value is 0.99, and I decided I was just going to buy a yacht and float away somewhere because this is remarkable. And that's my contribution to his story.
So does everyone clear about what a strain rate is? A strain is a fractional change in a dimension of some object which has a dimension, right? Like your heart muscle. And the strain rate is the number of seconds it takes to achieve that percent strain. So the units of strain rate are dimensionless because it's a ratio quantity per second. So it's seconds to the -1. So this is 1 over Bob's metric. Okay, this is in diastole. This is in systole. This is the log of the strain rate. Okay, and you see these numbers these lines are perfectly straight. I was able to get correlations on the order of 0.99 or better. So I think what I've got here is an epiphenomenon, which is kind of joined at the hip with the phenomenon of contraction and relaxation. And I'm proud of that. All right. Is there another slide, Larry?
I think you need to hire Banham.
Oh, Banham. Oh, well, who could be? All right. So let's see. Our feasibility study provides the required evidence that echo parameters of contractile and diastolic function are correlated with my metrics. By the way, I also did this in pigs with the special catheter that Larry alluded to, but that was long ago. And the next steps involve processing second studies data, which we're doing with medical students who are jumping up and down on a box according to a metronome, right? And we're going to be completing a study that will focus on repeatability of the metrics compared to the echo at rest. And we're going to be creating a real-time view and analysis of the Hirsch data stream. And that's all I got. Now I'm supposed to be in the OR.
Very good. You have a minute or two for questions?
Yes.
Okay, very good.
Just got 2 minutes.
Two minutes?
Yes, stolen time. Have any of you questions for me?
What's the name of the company that makes the small device?
It's called MoveSense.
Moose Head?
It's MoveSense. It's a Finnish company.
Oh,
You-
I don't mean it's
MoveSense.
ending. I mean, it's from Finland.
Oh, I know.
Right, yeah.
Moose.
MoveSense. And we know the engineers, they're friends of ours, they're friends of Larry's. And, you know, this is for $100 you can own one. And it was engineered to provide minute information about Olympians who do triple axles and this sort of thing. But it dawned on us that we could use it to get a seismocardiogram, and we were right.
One of the lovely things about this is Dr. Hirsch's intuition that these intervals could be put in diagnostic purposes kind of preceded the actual availability of commercially available to use. So as accelerometers got better, cheaper, smaller, it enabled a Finnish company that was originally a watchmaker make this device for exercise physiology. And then they have medical and can be white label. Basically, the processing of the hardware side of the equation.
Are you able to get it? What kind of intellectual property protection?
We have patents that are no longer pending. One of which is already issued, the other is still in the process.
Yeah.
There's still more intellectual property to be discovered and to be.
Yeah, we have a U.S. issued patent. We have several more after that. And there's a number of hurdles, even with this sense device in order to make it. And the FHA and JHF, Mike Wiley and his team and our innovation fellows, been very, very helpful in overcoming some of those issues. So we're looking forward in the coming year to also not just work on with medical students, but eventually with patients too.
Yes.
From a clinical standpoint, can you put this on just everyone with heart failure and it predicts who's about to go into a heart failure exacerbation, or like every pregnant person, and then it predicts who's
So
going to
I'm
go
glad
into
you
heart
brought
failure?
up pregnant people, women, excuse me. There's a mortality to go from being a pregnant person. There's a racial disparity in the mortality, 3 to 1, black to white.
Right.
And the reason is because these deaths tend to happen in the 4th trimester due to congestive heart failure. And the reason, Catherine, is because nobody's looking, nobody's monitoring. And it happens in the dark, in a vacuum. So you can monitor, you can have this talk to your iPhone. iPhone talks to cloud talks to doctors, and we can get people intervened with before awfulness happens. In people with congestive heart failure, in people who've been on ECMO, who've just recently been discharged, you can get them out of the ICU much sooner because you got this thing on there. You can see if they're deteriorating or not. You got a heart transplant, right? Is this thing working? Is this thing self-destructing? Is the patient's immune system eating it up alive on the inside? You can tell by looking at the numbers, and they can come right over the internet into your living room. It's not hard, and it's not even expensive. It's dirt cheap. It's the price of computing, which keeps getting cheaper and cheaper, even as I'm speaking to you.
I would sell it cheap though.
What? We'll sell it what the market will bear, but the point is, this can
She spent $800 for a sheath. I mean, for this.
This can decrease the price of access to healthcare, and it can make it more affordable.
You could put it on everyone who had a baby for these high catastrophic, low incidence events.
Yeah.
Because you could price it for that.
Yeah, and women are having babies later and later in life, and they have more and more comorbidities. It's not just about women having babies; it's about anyone who has congestive heart failure, or heart transplants, or massive heart surgery, or an aortic graft, a TAVR.
When are you planning to set up your company?
As soon as we can get the software doing what I just said it can do.
So
What we're gonna do, love is not enthusiasm.
I'm trying to figure out how I can give him $75K for maternal and infant healthcare
So-
grant that I'm going to be opening.
Keep me in mind.
So what we have to do is we have the data set, it's gonna be analyzed, then we're gonna do a repeatability data
Yeah.
set, because the FDA is gonna want this, okay? Then we're gonna go to patients, we're actually not going to the heart failure patient,
Yeah.
because sometimes their echoes are really ugly. We're going to MD Anderson,
Mhm.
where they get serial echoes every 3 months,
Which is brilliant.
okay?
Larry
So
thought of
once
this.
we have that, then we say, are we ready to raise our arms like a touchdown? Because the FDA, you know this, right? The bar to get a heart failure monitoring device approved. I was at Medtronic for 13 years.
Yeah.
How many hundreds of millions of dollars did we spend on one idea that failed slightly? And a device came out called CardioMEMS, and has run away ever since. So his enthusiasm is
Can't wear
important.
that.
The reality is you have to meet the FDA bar before you say you can solve the problem.
The other, as far as the startup company, a condition of the funding with the FHA is we got money from their venture capital group. We have like an initial phase, some studies we have to do in the coming 6 months or so, and then they have an option to license IP portfolio into the startup. You know,
So it's like about 6 months.
When's your grant money? Okay.
do it sooner the better.
But, but we run them on, but Mike, we run stuff on a cycle, and infinite maternal health care is one of the topic areas. And that same.
Every time I've looked at devices on the chest, there's a lot of noise. The second is, could you get the same kind of data from HRV?
HRV.
Heart rate variability.
No,
Are you sure?
absolutely sure.
So this is a very good point.
Yeah.
That he brings up heart rate variability, huge deal in the heart failure market. This is about electrical activity. There's no mechanical associated with it.
Yeah.
So if you want to talk about declining cardiac function, you gotta have the
You
mechanical.
can't tell just from the electricity what the heart is doing. You can have.
You would
can have-
likely
You
see
can have an EKG and you can have a dead person on your
a
table.
huge change in heart rate variability in the heart failure population, right, and in other populations.
Yeah, that's called association. That's a category in ACLS.
Many questions.
But
Yeah. Sorry, I got to Zoom. I'm in trouble. Thank you
Go
very
save
much.
somebody.
congrats, very good.
Question for Larry, and Larry can stay up here because he's got the next presentation too. Dr. Lombardi.
I think that was brilliant, by the way. Congratulations. That was fantastic.
I just came
I
on.
already thought of some applications for you to consider and something we can work with you on. And as we put all these devices in patients, they develop heart strain because we're putting these non-compliant devices in, and where the aorta is supple and retracts and contracts.
Your patients don't have
Not
subtle.
as much.
No,
not as much. But you could potentially predict those who are going to develop increased heart strain or a Winchester as a result of this increased non-compliance of the aorta with whatever compliance they have left, so to speak. So we can look before and after, and then develop some guidelines with respect to. Maybe
You're
these
gonna like
patients shouldn't
Rick's
get it.
talk. So he used the word Windkessel. So Windkessel is the German phrase for fire engine. So how did that ever come to be? So you had people in the community taking buckets of water, putting it in a big bucket, and it comes out in a steady stream. That's your Windkessel. Who knew you knew? Okay.
For repeat performance, yeah. Another section.
So I promise I won't have the same enthusiasm. All right. So this presentation was supposed to be between myself and Jeff. He's a cardiac sonographer and professor, but he's teaching a lab up in Worcester and he can't get out of it. So while I'm talking to you at the moment, as you sit here, your myocardial efficiency, that is the amount of work your heart's doing divided by the cost, is about 33%. The car is at 40%. Now, this metric, myocardial efficiency, has died away as a tool because it needed invasive tools. You needed to get coronary sinus stat, you need to get coronary sinus flow. Well, we've already talked about all this stuff is not happening. So I'm going to present to you today a concept, a non-invasive concept that gets us there. So why does this matter, right? So as you go into this disease states, you are in subclinical deterioration and nobody knows it. You don't know it and your doctor doesn't know it. Why? Not because they're not smart, but because they don't have the tools to look at any of this. And EF won't tell you if you're going subclinical. Strain may, and strain rate's not ready for Broadway. So as Dr. Lombardi discussing, as you get older, your aorta gets stiffer, so it dilates and it actually changes its composition. So it loses its Vinkestel properties. That actually puts a burden on the heart. It puts a burden on the brain, puts a burden on the kidney, makes your heart do more work. And okay, if your heart does more work, you're gonna have to consume more oxygen. So how do we get there? Simply, literally simply, right? So I've known about this for 35 years. But when I met Jeff at a seminar he was teaching, I brought this up to him. And 14 months later, we have a paper that's going to be submitted where we figured out a way to do this simply. So we are combining not brachial artery systolic blood pressures with stroke volume. We're using the SphygmoCor XL device from AtCor, where we get central aortic blood pressure, because we know that in the population between 25 and say 55, there's a big difference between your brachial artery systolic blood pressure and your central aortic. So for instance, me, I had my blood pressure taken the other day at my physical. My brachial blood pressure was 122/70. My central was 110/70. Diastolic's unaffected. So that means that every time you take a blood pressure with a brachial and depend on brachial, you would overestimate pressure in that population. So we're going to combine the central aortic pressures with echo to create the metric. And that metric is called myocardial efficiency. So stroke work, stroke volume times the pressure divided by the MVO2. And I'll tell you how we get there, because it is dang simple. All right. Who cares about such things? Well, on the left, starting in before 2001, these are the number of articles that were published evaluating aortic stiffness. So this was really due to the wisdom, foresight of Michael O'Rourke, a cardiologist who just recently died, and his colleague, Alberto Avilio. So they created a company after testing this against invasive measures. And what they've shown in this central panel is that you have two people with essentially the same brachial pressure. And here, the central and brachial are similar. Here, we have a really big difference. So this individual, you might say, is hypertensive, and this individual is not. That's all because of the mechanics of how the aging vasculature affects what's seen in the aorta versus what's in the brachial artery. So let's get into the details. So myocardial oxygen consumption has been around as a measure from the 1949. You can find the papers, there's not many, all the way to maybe 1995. And then they just started going to PET. So in 1982, Dr. Rook and Feigl did a dog study, and they created a non-invasive regression equation that only required that you have stroke volume, and we can get that from echo, LV mass, you have to have that from echo, heart rate, you have the aortic systolic blood pressure, you have body weight. So now you get estimated MVO2, and you never had to go inside the patient, and you can actually study this every time the patient comes in for an exam that includes an echo. So what do we have to do to prove that this is reasonable? Now, little segue. Dr. Rook was an anesthesiology cardiac fellow, and he published this, and I reached out to him 40 years later. And I said, hey, I'm using your equation. And he goes, oh my gosh, nobody talks about this anymore. So you're not alone if you don't think about this. So we measured in 8 normal subjects, we measured the variables needed to create the estimated MVO2. So 9.4 plus minus 1.6. So this is reasonable for the population that we had, right? Because the bigger the person, the bigger the stroke work, the bigger the MVO2. Thankfully, Van Oversheldt in 1993 did a PET study on normal subjects. And he came up with an MVO2 of 7.3 plus minus 1 and an efficiency of 35 where we had 37.9. But what is key about his findings is that the relationship between real MVO2 and estimated MVO2 was 0.92. So what we suggest today, and we're doing this now, and we'll continue to do it in all of the studies, is that a non-invasive assessment of this is easy to implement for patient care. So Dr. Cotto and I, we've already discussed this. How can we implement this in the women with hypertensive disorders of pregnancy? So with Dr. Mealy in our department. And then the bigger takeaway is that you can do this every time they come in if they're getting an echo. You gotta have the stroke volume and you gotta have LV mass. Now, LV mass isn't going to change really much every 3 months in the cancer patients, but in the beginning we're going to be very precise on this. So why is this? Right. So those are all easy. Those are really easy apples to pick. So you talked about, Dr. Hirsch talked about his metric in the hypertensive, in the pregnant population. So now we could easily add this to every population that gets serial evaluations. So we're going to start this at MD Anderson. We're doing this in the OR. I'll give a bit about that in a second. And then you have obesity and you have diabetes, as I talked in the very beginning of Dr. Hirsch's talk. So the one question you may have is, with disease states, what happens to MVO2? It doesn't change much. So as your hypertension gets worse and your stroke work goes up and your LV mass goes up, MVO2 will start to go up. But the problem you run into in an ischemic event is you can't provide the flow because the coronary microvascular bed's dysfunctional and can't auto-regulate from your local environment. So this is why myocardial efficiency starts to decrease. So this is a proof of concept study using the non-invasive central air blood pressure measurements combined with echo. And then we're going to go to all those populations we just discussed. And hopefully I didn't bore you or any other option. Thank you.
I'll throw one out. So as far as implementing practice, what SOPs and tools do you think put in place?
Yeah, that's a very practical question because we've been dealing with this as we step into the arena. Not all echoes are high quality, particularly in the strain world. It can get a little dicey. So doing an EF is really quite simple. Heading to strain is more difficult. Stroke volume means you have the parameters to give you the EF. So you should be okay. LV mass may be collected, but isn't considered as something they need to go quantify. And then the other component of this is that the at-core SphygmoCore system. So when you get a blood pressure taken in the doctor's office, they take one blood pressure. Well, there's no practicality to that being useful. Because I can tell you what we see in the OR, in the pre-op, of how patients' blood pressures are all over the place within 5 minutes of 3 measurements. So you got to get at least 3 measurements, and they have to be similar. And the machine tells you if the quality of the signal is high, and that gives you all these other metrics that I've not discussed. They're all useful, particularly for the patients who are getting in the aortic repair.
Any additional questions?
Thanks for listening.
Very good. So, thanks. So, and we had one additional technology to present, but I think Dr. Schercher is in clinic.
I just messaged him down a little bit earlier. He was going to come down at 3:30. I was trying to time it, right? So he should be on his way.
We had more questions for Larry than it would have all.
Or questions or
well, if
logical breaks.
yeah, if everybody wants to have a little bit of break or a discussion, wait another 5-10 minutes.
Can you tell me, what is the total volume of research that Cooper is doing with all of these great research institutes and things that you have?
On the innovation side,
Yeah.
the past 2 years, we've had 19 and 21 invention disclosures.
Okay,
Before the center started, we averaged about 3 a year over the
okay,
previous 10
but what's
years.
the volume? Like,
The dollar value of funded researchers, like only 4 million a year. So all of the inventions that you've seen today have come from unfunded research. And the initial funding on the innovation side, for 3 of them have come from the Foundation for Health Advancement, FACG, because they're all primarily clinically driven, so they're not coming from NIH fund research. But we have some of those too.
but is Cooper putting their dollars into for that? So if you-
Yeah, we
4 million external plus X from internal funding?
yeah, we have a commitment of just over $200,000 a year for an internal proof of concept fund.
Okay.
So we have some things like that on the health IT side in particular, where we have some projects with Microsoft. But it's a modest space as far as third-party funding. The nice thing though is that really, I think as Dr. Kirchhoff mentioned, the innovations are really problem-driven, so the solutions are quite practical.
It's inverted from the university model.
Yes.
These are very problem-driven.
Yeah.
Get the prototype going, and then hopefully the NIH, NSF, and other folks will see that value.
I would say we also have a good number of disclosures in partnership with Rowan University. So joint IP coming
Right.
from engineering and from the natural
So
sciences.
are
there.
your people considered Rowan?
I think everybody here that is speaking has a joint appointment at Rowan. I think where the intellectual property falls, it depends on who pays them. So usually anybody with an MD title is from Cooper, and usually anybody with a PhD title tends to be more from Rowan. Although I think we claim Larry Mulligan with the PhD is Cooper and not Rowan, right? You're the exception. You're a Cooperite, right?
I'm a who?
A Cooper employee, not a Rowan employee.
I'm a Cooper employee for all time.
But his intellectual property belongs to us because we pay.
And how many staff are part of the innovation? So part of the reason I'm asking is we run a website that's currently called Research with New Jersey, and we have information on Montclair, NJIT, Princeton, Rowan, Rutgers, and Stevens currently in it. We're looking to think about expanding, what's the base of innovators that we want to have searchable in that database. I'm just trying to get a sense as to, from here, how many.
You could include us. Our innovation office is actually bigger than the Rowan Innovation Office because currently we have 2 people plus Dr. Kirchhoff.
I want to know how many faculty innovators you have, because that's the number.
Like 20 invention disclosures last year. So as far as like hard metrics on innovators with proven inventions, it's about 20 a year. I think it's hard.
I guess
What's your question?
so, for example, if I look at Rutgers, they have like 5,000 faculty that are in this database, right? And if I look at NJIT, they've got, I don't know, maybe 1,000. So I'm trying to figure out like what's because we pay for each profile. So I'm just trying to get a sense. Like, is there
It might be
200
something we could
people,
look up
100 people?
with our research office, because we do have the Cooper Research Institute. And if the criteria are people that have, they're registered with the NSF and the NIH and the federal funding bodies, they could probably pull that information. I'm not sure what it
All right,
is.
we can talk later. Because again, I'm thinking, if we tell people, go to Research with New Jersey,
Mhm.
Go to that little search bar, type stuff in, you can find collaborators. I want them to be able to find these wonderful collaborators as we're trying to attract people to come to
Well,
New
if
Jersey.
you connect with me, then
Yeah.
I'll make a connection.
Yeah.
I don't know if you know Harry Mazurek. He was the director of the Research Institute here. He's now retired. They're recruiting a new person in.
Mhm.
Between myself and whoever they appoint there, we can pull that
Right.
information and
Yeah.
maybe get a list for you, because they definitely have a list of everybody that they've worked with that is interested in research.
Sarah Beth has a, oh, I just have a quick question. I guess this is for all the innovators. As you move forward from prototype to actually manufacturing and things like that, are you looking to manufacture in New Jersey? Are you looking to outsource? And the reason why I ask is, we have funding to help, manufacture and do it in New Jersey. I know it's a lot more expensive, but a lot of people that business I've worked with, they go to China or India. But I just wanted to see, have you thought about that, or what are your ideas?
If that's a possibility, I'm game.
Okay.
However, with the complexity of what we proposed, it's likely that we would partner up with an existing vendor that has great familiarity with designing these types of products, namely stent grafts, and those with the Dacron platform as well. So it's possible we may have to latch on that way.
Okay.
But it really just depends on how it goes in place.
And can I just add, because Synectic's here, Tim, you might want to chat with them and see if they'd be willing to, because they're working on a couple of these projects. But I'll just mention one of the consistent problems I think that we run into is, I say a lot of the development firms, not Synectic, they get this, but they can't speak alpha. They are so far advanced. And I hate to say this too, even contract manufacturers are used to working with really large players, the big med device companies, right? And so if you're a small company, we're working on a project right now. We're trying to get quotes from, and so if the state could encourage that in some way, I think it would open up, make it a little bit easier sledding for these early stage startup companies. We get it done, but it's all around the United States and, pressing in.
Does New Jersey MEP help you potentially identify people in New Jersey who may be able to do?
We've not gone down that path. We typically work with our development partners, like a Synectic probably knows a CDMO that's a step beyond, or they may have their own manufacturing capability. That's typically the way we go. And then, as Dr. Lombardi said, depending on the particular application, that kind of takes you to a particular manufacturer.
That's great. And I think we're also a stepping stone. I'm with Synectic, we're a design and development firm, but we're that stepping stone to get to the Johnson Valley, right? To go through. We have a quality system in place. We're documenting everything that we do, testing against the specification. We're able to measure and have clearly defined application. That will help them then get to the Johnson introduction.
Thank you.
And the only thing I wanted to add about the line of questioning going on is that sort of our office has three goals. One is to bring value to Cooper and our communities through commercialization of our own intellectual property. Two is to inspire entrepreneurship and to educate around value building within not only our own team members, but the communities that we work in. And lastly, partnerships. So what I'm hearing about from researchers and a database where folks can log on, what we're doing in the state of New Jersey in terms of development, et cetera, and also the money and development partners within the state. One of the things that we're very interested in doing is not only connecting clinicians with other inventors, et cetera, but to be a clearinghouse for, let's say, basic scientists at Rowan, Rutgers, and JIT that might have an idea and they might have an inclination. This may have a clinical application. We want to hear from them. We'll talk to them, put them in touch with our clinical expertise. And while we might not have an inventor, we might have a clinical expert that then can speak to this technology and does it have a pathway to the bedside that can bring value to the inventor and the communities we deploy it in. So it's a threefold job that we do. And if anybody has any contacts within the state of New Jersey, about databases, et cetera, benchtop researchers, PhDs, whatever they're doing. If you have a technology and you want clinical input, that's the third thing we do. And the only other thing I'll just say in terms of your questions about how much we have and all that, Dr. Lemon's been here for 2 years with an office of him and one other person. And he's accomplished through the great work of all our inventors this much. So if you're looking at, and if you're looking to throw money at an innovation center so we can do even more for the state of New Jersey, we're willing to take your check.
Yeah, I just like to kind of emphasize what Dr. Kirchhoff said about the external innovation side of things, because really we can put what we do in 2 buckets. One is the internal innovation and supporting that, which is what we're doing here. The other is working with companies like Johnson & Johnson or even other startup companies that originated from other New Jersey institutes or outside of New Jersey to be clinical partners for them. There's actually some New Jersey startup companies that we're currently doing that, offering clinical time in kind to their company. And we also have opportunities to put together, I would say, relatively innovative agreements so that we can be a part of their company. So like putting safe notes together for clinical time in kind as opposed to just making an investment in cash, because what a lot of these companies need are clinical partners and physicians that understand the space that they're working in. So, and that's another reason why the FHA has been such a good partner, is in making a lot of those connections with other companies that have been spun out that they support out of Princeton, Rutgers, and the like. So especially Princeton, which doesn't have a medical school. So we're very interested in partnering more with them and NJIT to be that clinical for their technology. I think we're still waiting, Chair, on Dr.
Yeah, I haven't gotten a message back.
So if you need a break, a minute or two, he'll come down. He's the last presentation of the session. But if he's stuck in surgery, then.
So he was just seeing patients.
Okay.
Step down, see if I can get hold of them.
It's an interesting website you have.
Oh, thank you. Yeah,
Oh, yeah, we do that also.
you should.
New company.
Absolutely. You're always going to right? Little half of the
$10.
Acela. Don't tell me how much it is when you try to go into it.
The thing is, actually, so on the pain side, Mike's been really good, and he likes. I've been trying to introduce.
You also with this?
Yes.
Okay.
Yes, Patrick. LSU, UCSF, and that or. Very engaging summits. Like that. Some used to form
Basically, what we do is
so there's some areas where
Maybe we can spend about 5 or 10 minutes again. We are like.
So basically, we ought to go.
Right now, just
Yeah.
covers our one and
Yeah.
considered an urban research. So I might be able to call you a new researcher using the new Carnegie classification.
it's because we have
Yes, as long as it's reported to NSF first.
hop on or contribute or take.
Yeah, well, we had super.
Good, yeah.
Yeah, we kind of got the.
No, because again, I can't just say, oh, I can't like, I have to be.
Everyone gets some reason.
Yeah.
Translational.
But on the other hand, I can't put like, because we pay $35 for the profile. You know, that's in the database. I only really want people who are doing research, you know, active researchers, who have like papers that are publishing. That kind of stuff.
Like we could create this.
Yeah. Yeah.
People are.
Right. Oh, yeah, so it's probably like a.
Yeah, I think that's pretty
Yeah, I guess I'm just thinking. And again, like, Rutgers is like. It's. I know.
So I would think.
Just,
Well, no, but our people are paid.
Yeah. I'm thinking of Catholic New York. You know, with that calamari. Yeah, but I thought she was the one who had the company. No, they had a company and then it kind of.
I'm not.
20 years ago, that was 20 years ago. That was like struggle, right? Struggle.
I do it at post-op. I would say like up to 4 hour check. Yeah, you can do it at a smash.
All right, okay. Again, when I just asked for these, I need to be able to, I got too much both times, $35. And of course. I gotta like knock my work cut out. Super.
Yeah. Thing is, it's like a drug. Disposable for a day.
I have 9.5 million this year. No one's in his proposed budget.
Entire patient population.
And go home. Forget, I can't add anybody. We don't care. I'm gonna need the legislators to like through that negotiation.
Oh, okay.
Kind of like from right now.
Maybe longer.
Like we are like employees, we want to get this.
I was curious because like your question is like how long is it? Right? Define like what is the reason? That's right.
That's right, you'd be like.
It's a big hit business.
One here, you are, what's your name?
The spectrum is, they want, you know. Or they require.
That'd be great. That'd be great.
I tried to get my. She cannot. Yes.
It does. I would say that it can be controller. I think
Where's the? Yeah.
This one on there.
If I was able to do one of these loud results, we have a mic. Thanks for coming. Hello, everyone. All right. All right. Thank you so much. We have Dr. Scherzer here, direct from Seeing Patients. Great presentation. This is one of the technologies that's further along. We have our prototype and proof of concept completed. And also another technology that with funding from the FHA. So without further ado, Dr. Scherzer.
Thank you, sir. I appreciate everyone's patience. I'm sorry that it was a little tardy today. You know, we at Cooper have a pretty high volume program across all specialties. We have a representation from our incredible surgeons there on vascular. They're a very high volume program. This is actually one of the unique opportunities that we have here, especially that we have inventors who are within our clinical ranks who can then apply those inventions directly to clinical care. So it's really an opportunity for us to take an idea and then take it through the full spectrum of activity all the way towards even clinical trial within our own institution. So that's really an exciting thing that we're going to continue to push forward. So this project is actually in concert with, and it's really 50-50 from a clinical standpoint with Wissam Abouzghib, who I do not believe was able to come today, but he is one of our incredibly busy interventional pulmonologists.
Sitting here early.
Oh. Well, there you go. There
He
you go.
was here early.
All right. All right. Well, you know, he might arrive at some point. So we do not have a good title for this, but we have called it the body cavity evacuator. So this is an idea that was born many years ago. Mike Kirchhoff was one of the initial ears to this idea, and Dr. Carpenter as well. And this was born actually prior to the pandemic. It was made fruitful during the pandemic, and then we drove it home with NJHF funding, which we are very thankful for. So there are many people that go into this, so really could not be done without Neil Lemon here. He was truly the glue that put it all together. We had the idea, we worked through IP, and then you really have brought us to a much better place and allowed us to really come together as a team. And now we're working as a bigger team. And we've got also Kelly Jang, and now we're starting to work with RQM Plus, Shamik, who is our contact for regulatory. So what is the obstacle that people in our space face, and what is the opportunity? So there are two issues that we identified as real issues that had an opportunity for a solution. One of them is called a pleural empyema. So an empyema is an infection of the pleural space around the lungs. It's a collection of pus in this cavity between two layers of pleura. It's caused by infection. During the pandemic, we saw a lot of viral infections that had superimposed bacteria. We had to deal with this a lot more during the pandemic than ever before. And there are about 32,000 cases estimated in the United States per year, and this is just in the US. The big problem here is that 20 to 30% of these patients affected will either die or will require further surgery in that first year of diagnosis. Many of these patients are older. So this is a pretty important issue that really requires pretty prompt multidisciplinary management. So right now, the methods of evacuating pus are largely ineffective, especially if patients come in a little bit later in the process. We put chest tubes in. That's usually the first intervention. Oftentimes, we put pigtails, small bore chest tubes. Sometimes we put larger bore chest tubes. But most of the time, these patients end up having to go to the operating room for either a minimally invasive washout of the chest or a thoracotomy that requires a big operation for a patient that's elderly. The second problem is a retained hemothorax. This is actually much we see this all the time, actually, throughout the United States. About 300,000 cases defined as a blood in the chest that is occupying 1/3 of the pleural space, cannot be drained by a chest tube, and that's there for 72 hours or longer. There's actually literature now in the trauma community that patients who have hemothorax actually are recommended to go to surgery earlier, usually within the 3-day window on presentation. So this is a really big problem. Why is it a problem? Ongoing bleeding, possibility for superinfection, but also most importantly, if you don't drain the hemothorax, a lot of these patients are young. They develop a rind around the lung that cannot be removed later, and they get significant pulmonary dysfunction that really impairs life. So prevention of fibrothorax is very important. Right now, the most effective way to do this is actually with VATS, minimally invasive surgery. So it requires a trip to the operating room. So thus was born this idea, especially, and it was really kind of materialized during the pandemic when we did not have all the resources to be able to take patients to the operating room willy-nilly. So we thought about this concept that we had been doing. We're putting two chest tubes into the space, irrigating, and we actually saw a lot of success with that, as many groups have in the past. But this hurts. It's two separate intercostal spaces. So what do we propose? We proposed potentially putting a tube into one intercostal space that is placed in using Seldinger technique. So it's a wire technique that any individual in the hospital that does procedures knows how to do. We do central lines this way, we do pigtails this way, and essentially this can be done in the emergency department, it can be done in the intensive care unit, it can be done anywhere in the hospital where you have a procedural space, or even in the operating room if you need to. And it's all done through a single point of entry. So every time you have multiple points of entry, you have multiple violations of intercostal nerves. And so that causes more pain. And these patients then splint and they have pneumonias. So really a single point of entry is very useful. We derived this for a population of 18 and above, although it can be considered for some larger pediatric patients, and that really is based on adult body habitus. We do not specify BMI. And this could be in a number of different patients, both trauma and non-trauma. And we talked about the conditions, and this is the IP initial design. So we have a full patent It's an international patent now as well. It's listed US patent. And the design is that we have a sheath through which two tubes go in that are connected, two pigtails in opposite conformations, one that's made for drainage, one that's made for irrigation. And we'll look at that separately. So with our help from our Synectic colleagues, we actually engineered these catheters. And so these are catheters, look, you can see on the top side there are two Wayne catheters that are in different directions, one that's 16 French and one that's 12 French. And they're put in through a guide wire. We also have this breakaway sheath, the 26 French inserter that we were lucky to find off the shelf, which actually makes it a little bit easier to produce this product and a little bit less expensive for patients ultimately. So we tested this. This is actually testing done by our engineers, and you can see that the catheters go through that pretty well. So this was all outside of or ex vivo testing. So once we did this, we built a model. So this was a non-animal based model simulating the lungs. And so on the left part of the screen, you could see these balloons that simulate lungs. And on the right part of the screen, we had saline in one side on the right, and on the left, we actually used Jello. So it's a more gelatinous material that looked like blood, right? Like coagulated blood. And so we tested this device using a number of dilators, and we sort of created our own little pathway here. So here's the way it inserts. What's interesting about this video, the first part with saline, we used betadine to see what the flow mechanics were, because we actually did not know how the flow would be through the tubes. And we wanted to make sure that whatever we irrigated through one tube didn't directly come out through the other, because that would be almost nonsensical. So we actually had an experiment to measure flow here, which seemed to work pretty well. It's showing video. And that's Dr. Wissam right there. Again, two tubes through one space, and you can see betadine flowing through one side. It's completely diffusing, while the other side is actually hooked up to a normal Pleurovac, minus 20 millimeters of mercury of suction. That can go up to minus 40. It took about 10 minutes for the betadine to start sucking out of the other port, which really was a wonderful finding for us. We did the same thing for the Jello side, and this was a test to see if the same concept could be applied in a more viscous state. And we were able to drain about 50% of that volume with Jello over the course of about an hour through this tube. So that was successful. So then we stepped it up, and we actually had this is a goat, the full thorax of a goat. On the right side, we have the goat intubated. So we actually simulated the way the lungs look, and we did a proof of concept. And I'm sorry if anyone's squeamish here, but oh well, we're all family here, I guess. Tasty. Tasty. Exactly. So anyway, so this simulates the actual procedure itself and how it can be performed. So you use a series of dilators over wire. The lung is inflated or inflating. So it simulates the actual model itself. Here we were seeing whether or not these dilators were going to be hung up, or there's going to be a lot of resistance in entry, because we want to make this as easy as possible for non-surgeons as well as surgeons to perform. This is that dilator, the sheath. We chose sizes not randomly. We chose sizes based on flow mechanics and ability to suction and irrigate. And we found that a 16 French tube worked a lot better than a 14 French, which is surprising that a 12 and a 16 fit through a 26 French sheath, but it did without any issues. So again, same idea. catheters coming in different spaces. They can be manipulated once they're through that 1 intercostal space, and they can be in separate areas to capture fluid. So our next study was an actual porcine in vivo study. So we actually got an approval through the IACUC to do 3 pigs to see if this actually worked in live animals. And we instilled 500 milliliters of viscous autologous blood from the animal, and we added 350 milliliters of saline to both pleural spaces. We waited 30 minutes for clots to form. The reason why we used pigs is pigs actually clot much faster than humans. And so the clot is much thicker, much more difficult to suction out. So if this worked in pigs within 30 minutes, it certainly would work in humans. So this mimics a hemothorax at around 48 hours. And we evaluated the pleural space with intraoperative ultrasound and chest x-ray for depth of fluid column. And then we inserted on one side a traditional 32 French chest tube, which is what we would do for patients in the trauma bay. And then on the other side, we put in our other catheter. So here on the left side of the screen, you can see the x-rays. So as before installation of any fluid. And on the right side, you can see the 2 blue calipers demonstrate that we actually did have blood that remained in the pleural space on both sides. So this was a successful hemothorax. We waited 30 minutes. We confirmed that blood based on bedside ultrasonography. And then we placed our catheters, 3 different locations for the 3 different pigs. And what was interesting is that, as you could see, all that blood, that column had drained out. It wasn't 100%, but it was enough to allow full expansion of that lung. So in pig 1, we had 25% less retained clot on the device side compared to the 32 French side. Pig 2 had about 25% less retained clot also on the device side. Pig number 3 didn't do as well, as 15% less retained clot. And we measured this by volume after we actually did the necropsy. The animal died, and we actually took the clot out and we measured it side by side, head to head, based on volume. So successful initial pathway. And this really completes phase 1 for us. We again thank, this could not be possible without the financial support of our partners. And we're now discussing the regulatory side. What would it take for this device to move through phase 2 and phase 3? And our hope is to engage all groups and promptly go into phase 2. I'll take any questions. I think that was 15 minutes, right?
Where would you get your prototypes manufactured for doing, to kind of move into the next phase of testing?
So that's again up in the air. We have great partners in Synectic, and we have the opportunity there. We also, this is something that we can actively discuss. Cost is going to be an issue. We have all the components that are off the shelf at this point, which is really a gift. We didn't have to mold anything. It's going to decrease the amount of time, the amount of money that it's going to cost to develop this device. So we actually retrofitted everything, and what we need to create is essentially a box, right? A set that is all sterile, and that really is the next step.
I would say from the kit perspective as well, Synectic has connections with other Matt, is it?
Yeah, we're owned by a company called Matt Holding, that's based in Vermont, and then we also have connection with Christian Hospitals, Martech, Medical, and
there's just some potential partners. But like Dr. Scherzer said, the individual pigtail catheters, we were able, there was a lot of searching done, but as far as the sheath and those catheters, I think there was actually only one combination application. If that wasn't the case, we would have had it with this. So lucky from that perspective.
And really that allowed us to go through phase 1 in a I'll call it a financially responsible way, where we can really focus a lot of our efforts on the engineering side, and then focus a lot of our efforts on the actual the models. Otherwise, we would have spent all of our money on the device.
I think it seems obvious, but this was all basically done Synectic support. Clinicians paying for it. The animal study was here with Cooper. The clinicians are
Right.
So they're compressing this development timeframe, I think, pretty extensively because it's such a tightly coupled. Shouldn't be glossed over that the good job that Cooper's going shepherding the project.
Going through the IACUC, through the use of the facilities and ordering the base foresee model, all of that's in house at Cooper. And we do have like a state-of-the-art facility.
And really, our advantage is that, and our second advantage is what do we do once we're done with animal studies, right? So we can do animal studies in our own facility. We have a wonderful vivarium that's very supportive, quick turnaround with the IACUC, and it's actually a very good IACUC. The other part, which is a gift for us, is that we take for granted as clinicians is we can actually apply some of this, if we get FDA approval, to our own patients, right? So you can essentially do an at-home initial clinical study that can then be validated at another center. So it really is, it's a unique opportunity.
Would you think, Neil, that they might look at some point to like federal SBIR?
Oh,
funding?
That's one of the steps. I know we're right at that point before phase 2 in the partnership we have with FECG to execute on an option on the second phase. And then in parallel with that, apply for maybe a phase 1 or phase 2 SBIR grant. That is definitely some interest in that, or also looking at putting a management team in place. I know Dr. McNacken as well in her technology has participated in I-Core, is pursuing that path as well. So definitely.
Because NIH also is doing some kind of free boot camps for companies to help them be first-time applicants to the SBIR, process. So that
I know one of the things I provide mentorship to the NIA, the National Institute of Aging, and they have a startup program in there where we partnered with one company to work with Cooper clinically, and we're hoping to have more. So if there is any additional partners with the NIH that have that sort of innovation arm with developing, I don't know of them. Please let me know. Because every opportunity will take.
Do you want to talk about the SBIR through CSIP?
So the Commission on Science, Innovation, and Technology has a suite of programs, grant programs, 25,000 up to 250,000. One of the programs that we have is if a company has received a federal phase 1 award within the last 2 years, they can apply to basically get an automatic $25K on top of whatever you got from the federal government. And then if you're in between phase 1 and phase 2, you can apply for $50,000. We changed it to a rolling basis, so there's not a strict window. We just kind of do a 2-year look back. We limit the number of SBIRs that a company would have gotten. Some companies get like 10, 15, 100. They're kind of like little SBIR puppy mills. We want to be supporting companies that are really using that as a mechanism to move forward. The Rutgers Small Business Development Center has a FAST grant from the federal government, and they're supposed to be available to help with registrations. And I'm trying to get them to have a little bit of dollars that might be available to help with grant writing support. They're going to be hopefully telling me what they're planning to do with that in the next couple of weeks.
No, we'll definitely have some candidates for that this year.
Great.
Some of them in this room, and then some in other areas as well. And then some also through external partners. Again, I know we work with Brittany Mason, that's early Alzheimer's identification. She's based in the university as well. And we've applied for grants with her as a partner for SBIR.
And then our just as an FYI, our research grants typically are 75,000. Some are like, we're going to do one on infant and maternal health care, and that's a super huge definition. Another one on food and agriculture innovation, one on sort of what we call catalyst, which is sort of everything else that's not clean tech, and then some that are clean tech.
And definitely, like we were speaking about, like, Dr.
Yeah.
And then potentially even applications for Dr. Mulligan's second. No, sorry, Dr. Blake.
Yeah,
Yeah.
your programs sound fantastic.
They are fantastic.
And
That's why he's gonna move here, find him space, South Jersey Tech Park maybe, or here.
So yes, just as a coincidence, yesterday I was speaking to Stephanie Davis, who runs the SBR program for national lung, the NIH program for lung,
Yeah.
blood,
Yeah. Mhm.
something else. And Stephanie has 130 million. 10% of NIH is $1.3 billion. So they have a huge amount of money. And I'm very interested in seeing the companies that they fund get into the market. Very interested in that. I think that's why she was talking to us.
Mhm.
But I wouldn't wait. One thing we do is we go early. We form a company early.
Right.
We go to the FDA early. Just do a Q-sub, doesn't cost anything. And then go for SBR funding as early as possible. If you have a company, you have a vehicle that can accept funding. Sometimes you could turn down a lot, but you can go back and you can talk to them. They'll help. It's a fantastic program.
It's like billions, they give out billions each year.
That's non-dilutive.
Non-dilutive funding.
But they have special programs. One of our companies got a $5.4 million SBIR grant. It's called the U44.
Mhm.
5 years, $1 billion a year. Worth having.
We definitely, our startup companies will be focused on problems and looking, when I was at one startup company that is focused on flaxseed lignans. It's an anti-inflammatory, and over 5 or 6 years, they've raised 13 million in different SBIR grants. They just keep going back and say, "Oh, does it work for this? Does it work for this? Does it?" But they wouldn't follow through with any one indication to the end to get into the clinic. This is where I think the technologies from MDC Studio and the technologies here at Cooper are really driven by physicians looking for solutions for the patient. So they would be a stepping stone. So we should have some applications.
And I know that NIH sometimes for companies who have gotten SBIR awards will pay for a company to go to the big bio conference. There's usually a section on the floor, which is the SBIR awardees. So again, that's an opportunity to like mix and mingle with 20,000 of your favorite friends and family.
Well, absolutely. Good meetings there, JP Morgan. I'm like I said, I'm hopeful that over this year and next, we'll have at least, we're also finding ways to work with other groups like Gila at MDC Studio. There's some additional areas on the emergency medicine side where there's a lot of opportunity in surgery.
Are you guys going to be part of our New Jersey Pavilion?
Yeah, we can. You can reach out to us. Also,
No, that is like the one of the
Gil knows, he can't get rid of it.
so through Camden and also with Rowan, there's additional partnerships and stuff that we've been able to do. So one way or the other,
because you help us sell New Jersey, right? Right. You help us attract people to come to New Jersey.
I'm definitely interested in that. And like you said, like the programs that both of you offer are special in the bigger context. So very generous and a lot of people that are very, very smart behind it.
The other thing I want to that Sarah Beth can talk about is golden seeds, which is the funding specifically targeted for any of your ventures that have a
I
female
mentioned
lead.
this to Dr.
Yeah.
McDonald, the Golden Seeds Funds, a good one. And also I mentioned Brittany Case.
Yeah.
Women CEOs.
Yes.
And there's also like programming through the university's center.
Yeah.
On the other side of the river.
There, and some of those, I think the program at University Science Center is part of NJ Accelerate.
Okay.
So if companies participate in that program and then they are doing stuff in New Jersey, there's some financial benefits. Not my program, so I don't want to 100% speak to it, but there's like a number of different incubators and accelerator programs that are part of NJ Accelerate. And I believe one of the Science Center's accelerator programs is part of that.
Well, I went a little long today, but I think it was well worth it. So thanks everybody for attending. We'll reach out to you. I think if there's any questions about working with the Innovation Center, I'll provide you my business card as well. And happy to follow up. And thanks for the first cardiovascular symposium.
Great.
Next year, alums.
Okay.
Someone had a lot of snacks.
Another name for that.
I need to stay up. I like another name.
Well, I.
University of Maryland representing here.